very much to Geraint for soldiering along in the lockdown and organising the seminar series. I think it's really valuable. And congratulations to Dana McGregor on making so much progress so quickly on the blackgrass. Looks like a tough subject, but one where every farmer will be waiting with bated breath. So I'm going to talk about some of our work today on assessing the effects of climate change on crop yield in the UK. This is work that we started recently since we joined the John Innes Centre, and it's really started to change the way I think about quite a lot of aspects of plant biology that I thought I understood quite well. So I hope you're interested in the sort of technologies I'm going to show today. I mean, you might not have your own farm, or you might not have some of the technologies here, but they're all available to the community in the UK through collaboration. So if you like any of them and you think some of them could be useful for your research, then please don't hesitate to get in touch. So we all know the issue in that we're in the era of global environmental change. And those of us who are outside a lot, which includes most plant scientists usually connected to the environment, will know that the country has warmed a bit and the weather's changed quite a lot already from when we were younger. And so we're already living in an era of climate change. And it's interesting to think about how it's going to affect agriculture and how it's going to affect our cropping systems. And when we look into the future, I'm particularly, you'll see why in a minute, I'm particularly interested in winter temperatures. There's really a wide range of predictions what's going to happen in the UK when it comes to temperature, when it comes to rainfall. And you can see here various emission scenarios for the latest UK climate change projections, which were run in 2018 by the Met Office. The top is the most pessimistic emission scenario, and at the bottom the most optimistic, which is basically we stop emitting carbon in about 2 months' time. So we'll be fairly sure that one's not going to happen. And you can see the types of winters we can expect. And then on the left, the temperature. And you can see that in the most pessimistic scenarios, about 10% of the winters could be more than 6 degrees on average warmer than the winters we get now. And even in sort of mid-range scenarios, a lot of winters are going to be 2 to 3 degrees warmer, and maybe up to 5-6 degrees warmer. And wetness is another prediction. I'm not going to talk about rainfall today, but rainfall is obviously very important right now because a lot of winter wheat hasn't got in because of the wet autumn. So all these changes are going to have effects on how we run our cropping systems in the UK. And if you think this is something sort of academic for the future, I'll take you back to this temperature series from December 2015. So December 2015 was exceptionally warm. In fact, we had a kind of weather synoptic setup roughly similar to what we're experiencing now. And we had these sort of southerly, southwesterly flows for the whole month, and various days were between 2 and 10 degrees warmer than average. So this type of weather, this type of warm winter weather that one would normally expect from sort of coastal Spain or Portugal, this can happen here already, and is predicted to get more frequent in the future. So how do we go about thinking about what is happening and what will happen to crops and the way they perform in the field, say for instance later this century? And many of you will be familiar with this type of research, but if you're not, essentially there's a large global industry which involves in taking global climate models, using them to predict what the weather will be like, and then hooking the outputs up to inputs from various crop models, and using these connections to predict what will happen to, for instance, to crop yields. And if you're interested in this type of work, there's a really nice example of this published recently from Caroline Harkness and colleagues at the University of Reading and Rothamsted. You show what will happen, what the most likely scenarios are for UK wheat production later this century. And really, these are the sort of gold standard approaches we have for understanding what we might need to do to adapt our cropping systems to climate change. But of course, as scientists, we know that there are uncertainties in all these models and predictions. And there are two major uncertainties. The first really is with the interaction of global climate models and regional climate phenomena, such as the North Atlantic Oscillation. We really don't know how these will play out in the medium term and what effects, therefore, it might have on our weather. And we have predictions, but we sometimes don't know how good those predictions are. The second area of uncertainty is that we know a lot about weather, and we know a lot about crop physiology, crop developmental biology, and crop yields, but we don't know what we don't know, essentially, about how weather affects crops. And there may be considerable number of important processes which are just not represented in our current crop models that might be important for yield formation. And if we're missing any of those, then there will be uncertainties in the outcomes. And really, that's the topic for this presentation today, is whether there's some key processes that are missing from the models. And what I'm going to talk to you about is some work we've been doing on winter oilseed rape in the UK. And it's a very interesting crop for thinking about the effects of weather on yield, because the yield is highly variable year on year. This is UK-wide data from DEFRA. And you can see that between different growing seasons, the yield varies from about 3 to 4 tonnes per hectare. Now, 1 tonne per hectare is worth about £340, give or take. The price obviously varies from time to time. So this yield variation is worth about 200 million to the UK economy. So think about what the weather's like, and the different weather scenarios and different growing seasons can have a big economic impact on our farmers. And we don't really know why this is. We know something about it, but we don't know why there's this large amount of variation. So the way we're coming at this, if we want to understand how climate change is affecting oilseed rape yields, we need to understand how the weather today is affecting oilseed rape yields. If we do that, then we can understand how changing in that weather might affect yield in the decades to come. And one thing that you need to bear in mind, and I'm sure most of you are familiar with this, that winter oilseed rape is a crop that's sown in August or maybe in early September, and it has an obligate requirement for vernalization. So the crop will flower in the spring, is grown as a winter annual. You do get spring oilseed rape, and it is grown in the UK, but it's a small area and the yields are much lower than in winter. And in the Brassicaceae, we know that vernalization behavior is governed to a large extent by a single gene known as flowering locus C. And you don't need to know much about flowering locus C for this talk, but you do need to know that flowering locus C expression starts off high and gradually is silenced during periods of winter cold. And this silencing is stable even if plants in the laboratory are subsequently moved into the warm. Now, in other species, there may be other genes that are important in vernalization control, but in the Brassicaceae, it's primarily flowering locus C. But look, control of flowering time is not the only function of flowering locus C in plants. We also know, and I can see that Gabriella Alca is here, so it's good that I'm citing your paper. So here's an experiment on the right from Gabriella Alca in Kathleen Donohue's lab, showing that if you increase the vernalization across a wide range of Arabidopsis successions, you reduce the seed dormancy. And our lab and Kathleen Donohue's lab have published several papers showing how FLC is also important in dormancy control, and that'll become important later in the talk. So, to start the story about yields and how they're affected by the weather, we first just took some simple analyses looking at high and low yielding years and asking the question, what is different about them? And this is a very simple analysis with a very simple linear model. And some of the key conclusions are in the chart on the left that you can see. So the black years are the highest yielding years, and the grey bars are the lower yielding years. And we're looking at the temperature in different months. And you can see immediately that the highest yielding years are characterized by warmer conditions in October and cooler conditions in December, and a sharp transition between warm conditions and cool conditions. Whereas for the low yielding years, you can see there's a more sort of bowl shape effect of change in temperature over the winter. Now, if we drill down in a bit more detail, not just looking at months, but looking at individual overlapping 20-day windows, which is what we see on the left. So where the blue line is above the black line, this is a positive relationship between temperature and yield, and where it dips below, there's a negative relationship. So in October, there's a weak correlation between yield and warmth, but then in December, we head into this period where there's a strong correlation between chilling and high yields. And of course, as that we've been interested in FLC for a while and its response to temperature, initially we started to think maybe this was something to do with the vernalization response, but we don't know. We could also see this effect in different data sets. So at the bottom, we could quantify the effect of a 1 degree Celsius temperature change in December on yields on farms in the UK. And each degree Celsius drop results in a yield increase, roughly we think of about 100 kilograms. And we also analysed about 7,000 yield measurements from the HDB recommended list and got pretty much the same answer, that a fall in temperature of 1 degree gets us on average about an extra 100, 110 kilograms per hectare of oilseed rate, which is not an inconsiderable difference when the temperature in December can vary between maybe 5 or 10 degrees in different years. So it's all very well showing that temperature has an effect, but we know that correlation isn't causation. We know that there may be other variables aliased with temperature. We know that temperature might affect physiology or development, but we also know that temperature will affect the activity of pests or diseases. It might kill insects, or something about the way it makes the plants grow might make plants more resistant to pests or diseases, or it might affect what a farmer can do on his or her farm. For instance, whether they can get on the land, whether there's still enough for spraying, and so on and so forth. So we need to start to design some experiments to address this. So the sort of next section is a story about how we convinced ourselves that this was something to do with the developmental biology of the crop. So the first thing we did at John Innes Centre is just have a look at what the crop is doing at different times of year. And Carmel O'Neill in my lab immediately found out something quite interesting, in that exactly at the point where the temperature relationship with yield goes from being positive to negative, is exactly the time of year where the plants go through the floral transition. And you can see this in the left. You can see the date here. This apex was taken in 3rd of November 2017. You can already see some floral organ primordia, and you can see how the inflorescence develops through November and into December, that growing season. So we think the plants are going floral, and that this transition between warm and cold happens at the floral transition of the crop. So the question is, of course, this could be a coincidence, but it also led to the question of what, therefore, is controlling the floral transition at this time of year. And to address this, we just did some simple transcriptomics to see what was happening. And you need to know that there are up to 9 copies of FLC in Brassica napus. And what you can see them do is interesting. So red indicates high expression and blue indicates low expression, and the dates at fortnightly intervals are across the top here. So you can see that the expression of FLC starts out high, but really by mid-October and the end of October, the expression of 5 of these FLCs is pretty much gone already, and it never comes back. And there's 2 other FLCs here on chromosomes C2 and A3. These take much longer to vernalize and actually don't vernalize away until after the floral transition, and that'll be important later. So we came up with a hypothesis that during this October period where warmth is beneficial for yield, this is when actually the vernalization that people have studied in the lab is happening in the field. So it's not in winter, but in mid-autumn. And the question arises how we can prove that. And the way we address this is simply to take the following approach. Now, if you apply warmth to plants, they tend to grow faster and flower earlier. But if you apply warmth to a plant that's in the middle of vernalization, it has the opposite response. So if you apply warmth to a vernalizing plant, you actually delay vernalization and delay flowering. So that's what we've been doing. And at the farm at John Innes Centre, we've been setting up field trials where we've been actively warming different field plots using cables on the soil surface next to the plants. So you can see the difference it makes here with this thermal imaging camera image. I think this is from December 2017, and then there's another larger field trial which we conducted the following year, and you can see a picture of our nice field station, which any of you are welcome to come and visit once the pandemic is over. And the first question we could ask is, what's happening to FLC? Is it, for instance, is expression being affected by warming if we apply it in October? And here's the FLCs, and you can see there's two copies here, the A3B and the C2. Their expression is not significantly affected by October warming, and these are the two which vernalize later in our original data set, as you can see on the right. But you can also see there are these four copies here, and their expression is much higher at the end of October after the heating treatment than after the control treatment. So the control treatment has vernalized these away, but the warmth has prevented that. And this, I think, is a first indication that these plants are really responding to temperature at this time of the year via FLC, and then they're going through the floral transition. And we can see that here in the unheated plots. This is UH. And in the heated plots, these are the apex of plants that we've dissected from the field. And you can see they start off looking nice and vegetative in October with these typical leaf primordia, the triangular shape. And then they go into November and they start to make these floral organ primordia, which start to develop through November and into December, as in the year before, which I showed you previously. And then the heated ones, you can see in November, these are still vegetative even towards the end of November, and only at the beginning of December do we see a floral transition. So heating has delayed the floral transition by up to a month, actually, this field treatment, which again shows that what's happening here is that the plants are vernalizing in October in mid-autumn and then going through the floral transition. But much to our surprise, delaying the floral transition by 3 to 4 weeks had a much smaller effect on flowering time itself. And if we look at some drone shots of a field trial, and I've just cut out the plots here, as you can see, the peak flowering of the control plots was around the 25th of April, but the peak flowering of the heated plots was roughly about 5 or 6 days later, the first beginning of May. So even though we delayed the floral transition by 3 or 4 weeks, there wasn't very much delay to the flowering of the plants. Now, this type of effect's been seen before. For instance, Johanna Schmidt's lab spent a lot of time growing flowering time mutants of Arabidopsis in the field and showed that they make remarkably little difference to when plants actually flower in the field, even though the plants are quite strongly affected in laboratory experiments. So this type of result is not without precedent, but it tends to suggest that there's something else affecting the flowering time of the crop. Now, for some reason it's complaining at me. So fertilization happens in mid-autumn, immediately precedes the floral transition in UK oilseed rate. And we think because cool Octobers are historically linked with lower yields, it's possible that premature vernalization is part of this. But there's something happening after the floral transition, which is just as important for determining the flowering time of the crop. And the next bit of the talk is really about what that is. Now, about this time, a very pesky pest known as the cabbage stem flea beetle brutally began to attack the John Innes farms. It has farms all over the southeast of the UK, and basically renders incapable for a couple of growing seasons of running field trials. And we turn to a different technology. We have what we call these smart controlled environment rooms installed at John Innes from Convirant. And they enable us to reproduce whole growing seasons at 15-minute resolution, starting in August when we would sow the crop in the field, going right through to July when we would harvest it. So we can go through through a whole growing season, simulate the temperature in the lab, simulate the changing day length, nightfall. We can do humidity if you want. But then we can also tune a cabinet next door, essentially to run a heat wave. And we can time the heat wave to exactly the time of year in the field where we know that temperature affects yield. So we know statistically when warm winters are associated with yield. So essentially program in a warm winter here, move the plants to a second cabinet, and then at the end of the heat wave, we move them back and they finish the growing season together. And there are advantages as well as disadvantages. You think maybe it's a disadvantage to come in from the field, but there are some advantages. We can exclude pests and pathogens, and we can vary the temperature without varying photosynthetically active radiation or water availability. Now, so if we see an effect, we can essentially demonstrate that it is a temperature effect and it is directly on the crop, and it's not to do with any of these potential confounding factors that are out there. So if we warm during December, after the plants have gone through the floral transition and they're just stable, the plants don't regress, we see some quite surprising things. The first thing is that warming in December after floral transition still delays flowering of the plants. And this initially was a kind of a surprise to us till we thought more detail. And warming also causes an increase in the level of the two FLCs, so in the blue and orange here. In the cold treatment, the expression is low, but the warming in December actually causes the expression of these two FLCs, which silence in winter, to stay high. So warming in winter causes these winter-expressed FLCs not to be silenced, and that coincides with some late flowering of the crop. So what we think is happening in this is kind of a standard model of how winter annual behaviour is controlled that you see often in the literature. The plants grow in autumn, they vernalize in winter, and then when it warms in spring and the day length lengthens, the plants transition to flowering, bolt, and we see the flowers. This is not what we think happens in oilseed rape at all. So what we think is that there's a two-stage process, and actually the plants vernalize in response to autumn cooling, and plants detect autumn. You can see it now, the leaves are falling, and we think that this process which we're investigating in the lab in Arabidopsis is probably conceptually similar to this autumn sensing process in trees and so on. Then in winter, they enter a kind of quiescent phase, a dormant phase, and we know that the flowering time genes such as FLC can regulate dormancy because we've shown it in seeds, as have others. And then if you warm in this period, of course, you prevent bud break, and then in the spring, the warmth and the long days, once the buds are not susceptible to dormancy, then they will come out and bolt. And this is how we think that the phenology is controlled. And it's much more similar to perennials or trees than perhaps we first realized. The December warming also has other effects on the crop. You can see at the bottom left, makes abnormal flowers, that delays the maturation of anthers, and delays or even inhibits the production of petals of some flowers on the primary raceme. And this is accompanied by big yield deficits, especially on the primary raceme, and lots of other abnormalities. And we've run this experiment twice. We've seen the same thing both times, thankfully. The heat wave in winter, then there's no effect on the 1,000 grain weight, but there's an effect on the number of pods which are filled, and an effect of a halving in our experiments of the seed yield per plant. And we see this in two sequential 10-month experiments, which are completely independent. So we think this winter warming, we can show that it affects yields and is a real phenomenon. So to conclude, we think that early winter warming is a threat to UK winter oilseed rape yields through disruption of floral development and reproductive development. And we think the warm winters are inducing a bud dormancy, much like you can see in perennials. And this process is what leads to the abnormal flowers and low yields. We know that there are these two copies of FLC which are temperature responsive there, but we need to do some more work to show that these are really whether these are really driving the changes in floral development that we're looking at. And that's the type of work that's going on in my lab now. So I hope you enjoyed the seminar. Please get in touch if you're interested in any of the technologies or using them, because we're always open to making them available to the community. I'd like to particularly acknowledge Carmel O'Neill and Zhang Liu in my lab, who have been important for the practical work that I've presented today, and also Rebecca Beebe, who's now at Helsam Seas, and James Brown for their help with the statistical analysis, which we also published. And, so thank you for your attention. I hope you enjoyed it, and I'll hang around with some questions if you have any. So back to Geraint.
Thanks very much, Steve. That's excellent. Really good to have some real-world consequences to what we're seeing there. So once again, encourage people to ask questions into the question box. Let me kick off though. So do you know? I'm sure you do know, but are seed companies already beginning to select for lines that don't have this yield penalty with a warmer winter? Steve, do you know if people are doing that already?
Well, it's quite hard to select for. I mean, you can argue that the breeders will select for it over time because the warming is gradual and then the new varieties are already being trialled. But yes, I think there are certainly some breeding programs that have an advantage in the UK climate, and a lot of breeding is done by continental firms, and they'll bring over varieties to trial in the UK, and they work great in Germany, Ukraine and Poland, where winters are reliably cold, and they bring them here and they don't do so well, so they often struggle. And you see that their successful varieties are often from their French breeding stations, for instance, where they might also be breeding varieties for, you know, the western fringes of France, which have got their own kind of interesting climate. So it definitely affects the success of different breeding programs and the products that they bring to the UK market. There's no question of that.
Mm-hmm. Okay, so we've got a question from Gabriella, who you mentioned in the talk. So she says, nice presentation. So was the seed quality affected in addition to the yield by the warming environment?
Thank you, Gabriella, for the question. You won't believe it, but we didn't even test this. We were so maxed out testing all the other things and trying to get huge numbers of oilseed rape plants and processed all the time that we haven't even looked at the quality of the seed. So unfortunately, I can't answer your question, but we have done other experiments that show that Brassica seeds are affected by all the same environmental condition, the variation environment, for instance, that you would see in Arabidopsis. So we have every reason to suspect it should be the same.
Yeah, just to follow up on that. So you showed early on in the talk, there was just that weak difference in the flowering between the ones which were delayed earlier on in the October. Did you see any yield change in those at all between those?
No, we didn't see any yield change in that experiment. And it's really difficult when you're in the field, because obviously what you'd really like is a growing season which is like some cold October from decades past, which you can then warm on top of. But you can't order like a cold October on demand from God. So we had a warm October and we warmed more, and I think perhaps there's a limit to how much yield you can get from warming. But yeah.
Okay, so a question from Jose Guterres Marcos. So, hey Steve, do you think these FLC genes in Brassica play different roles, and have any of them been selected by breeders?
Yeah, it's a good question, Jose. Yes, so we know that there's variation in these genes between different varieties of oilseed rape, and other groups have also shown the same thing. So we know that they're being selected for. We know, for instance, of varieties from China where they have a very minimal vernalization requirement. Incidentally, they also have this warm winter effect in China. We know that they have different combinations of FLCs, and that different winter varieties from the UK have different combinations. But what no one's really looked at before is how many of these FLC haplotypes are still going strong in the floral buds, because no one's really thought that it was important to look after flowering before.
Okay. So question from Fiona Cork. She says, Steve, when warm December reduces the flower number on the main raceme, do you get increased production on the side branches?
Right, that's Fiona. Hello, that's a good question. So, yeah, maybe I was slightly misleading with what I said. It doesn't reduce the flower number. In fact, it can even increase the flower number on the primary raceme as the plant tries to compensate for the fact that its flowers haven't been fertilized. It will go and make more flowers. So the answer is that some varieties will make more flowers on the primary raceme, and some varieties will turn to the side branches and make more flowers on the side branches. So they will always try and make more flowers, yeah.
Okay. So a question from Pete Whaley in Liverpool. So he asked a question about the diversity set. Have you had a look at any variation in response to temperature in the Brassica napus diversity set?
So that's a good question. In fact, I've got a very hard working and hard pressed PhD student now who's in progress of exactly that experiment, and he has the unenviable task of taking varieties with all sorts of different flowering times and trying to get them to all go through the floral transition at exactly the same time in autumn, and then expose them to different temperature treatments. So if he's successful in this somewhat ambitious endeavor, then we might have something to say about that in a year or two's time. Okay,
one for the future there. Okay, so question from, oh, they're coming in fast now, that's good. Another question from Gabriella. So would you expect different effects if you would apply the warming environment at different moments of the day, for example, warm nights?
Yeah, it's a good question. The reality is that we would love to do all these different experiments, but we simply, well, the bottom line is they're very expensive. So running these cabinets is costing us £300 a week. So we need more funding to try these. But yeah, so I'd like to know the answer to that, but we simply can't do the experiments right now to tell you.
Okay. Okay, question from Wim Sop, and they say, what is the evidence that the floral buds are dormant since they grow during cold exposure? Right, that's a good question. So there's a common misconception that dormant buds do not grow, and there isn't a single perennial species I know where dormancy is allied with complete non-growth. So even in a dormant tree bud, you'll see some growth. So I wouldn't necessarily say for him that this is exactly the same as a dormancy program in trees, but it has the sort of similar properties in that the warmer the buds get, the later the bud break, and the colder the buds get, the earlier the bud break. And that's what the main similarity to bud dormancy is. Additionally, I haven't had time to show it today, but if you warm the buds, they accumulate abscisic acid in exactly the same way that you would expect during a bud dormancy program. So this is why we think it's kind of a dormancy phenomenon. But if you were trying to say that maybe it's not exactly the same, then I would completely agree. Okay. Okay, so final question. This is from Steve Puttit and asks, is fertilization within a salique reduced by the warm winter period?
Right, yes, it is. So exactly the nature of the fertility defect is essentially the most active topic under investigation in the lab. But these experiments, as I'm sure has happened in many of your own research programs, have been disrupted a lot by the lockdown, and I'd hope to have more to say about that. So essentially, check if you're interested in that topic. Check back with me in a few months, and I hope to have something that I can say with more certainty.
Okay, great. Okay, so I think we'll leave it there. Thanks so much, Steve, for speaking today, and